Elisa can be used on a wide range of different sample types based on what exactly we are set out to do. Elisa is done on micro-tidal wells. There's usually 96 of these wells arranged on a plate like this. The advantage to having our wells arranged like this is that usually for a quantitative assay like ELISA, we would be working with a range of different concentrations, and ideally we would also be working with a number of copies of our sample and sometimes different sample types. So it really helps to have our wells arranged like this because it makes pipetting so much more easier and the whole process just more efficient. And our wells are marked numerically on one side of the plate and alphabetically on the other, which should remind you of a chessboard. Okay, so if we wanted to do indirect ELISA, these are the steps we would follow. The objective is to identify or quantify, usually quantify, an antigen that's marked here in red. So we would coat the inner surface of our microtiter well with our antigen, and then we would add the primary antibodies. We would add the serum that contains the primary antibodies and then wash off any excess that is unbound. And then we would add the secondary antibodies and wash off any excess. The secondary antibodies are bound to an enzyme. This enzyme, when it encounters its substrate, produces a distinctive color. So since the primary antibodies bind to the antigen and the secondary antibodies bind to the tail of the primary antibodies, when we add the substrate, based on whether we see a color, we can tell whether or not the antigen was originally present in the well. And based on the intensity of the color, we can do some calculations on how much of the antigen might have been present in the well in the first place.